Hey everyone, it's Ben and Brooke here. We're going to be taking the time to take you through our journey of exploring the function of our gene assigned for this semester in Brachypodium distachyon.
The first way that we analyzed our gene was by using bioinformatic tools like FNesh, which is an ab initio model, and Phytosome, which is a homology-based model. This is possible because there's a lot of publicly available data on the distachyon genome that our gene can be searched against to predict things like the protein coding regions, introns, and the protein sequence that's encoded by the gene. And all of this information is available because DNA sequencing is now cheaper and easier than ever. So there's a lot of ways that you can analyze sequencing results, but here we're using an ab initio model and a homology-based model.
The ab initio model by FNesh predicted from our gene sequence that the gene has a coding region of 490 base pairs, spanning from 650 base pairs to 1,140. It is also indicated that our gene contains one exon and no introns. There's also a large 3' untranslated region before the poly A tail.
Next, we searched our gene against the entire distachyon genome in Phytosome. Phytosome assembled a full EST or express sequence tag with only one exon and no introns derived from cDNA clones. So an EST is assembled from Phytosome's library of cDNA clones, which are copies of mRNA, and a sequence is produced. The log scale RNA-seq coverage is shown in blue, where the height of the peak describes the relative amount of sequence that was found to match the transcript.
We compared our protein sequence predicted by Phytosome to similar sequences in other closely related plants like Oryza, Ambarella, Arabidopsis, and Solanum. We found that the closest related protein to ours, indicated in the red box, is an ATP-dependent RNA helicase protein in Oryza sativa. We then use this to compare the expression of this gene in Oryza to the expression of our gene in Brachypodium.
Next, this gene expression heat map from Phytosome shows the relative expression of our gene, which is relatively low overall, but which is indicated by the yellow color, but is relatively high in the shoots of etiolated plants, as indicated by the red color. Etiolated meaning that the plant seed have never been exposed to light of any kind. And then we've next found that expression of our gene is highest in the stem base as compared to the stem and stem tip. This is also seen in the figure on the right, which shows the highest expression early on in the base of the developing stem.
The next way that we analyzed our gene was by investigating the effects of a mutation in our gene. We grew wild type and mutant Brachypodium distachyon plants for this analysis. We extracted DNA and amplified it for sequencing to confirm the presence of said mutation, and compared wild type and mutant phenotypes to each other.
So using Phytosome 12, we identified the exact location of the mutation so that we could design PCR primers that would amplify the mutation. At position 919, a C nucleotide was mutated to a T, resulting in a stop codon gained. The log scale RNA-seq coverage is shown in blue, indicating a halt in expression at the location of the stop codon. This is the only high impact mutation that we found. The others were low impact.
Once we found the mutation that we wanted to amplify, we extracted DNA from our wild type and mutant plants. These are our quantification results from molecular absorbance spectroscopy, which is nanodrop, and fluorescent dye quantification. The concentrations differed because the nanodrop detects all nucleic acids and cannot distinguish from DNA and RNA. The fluorometer, however, uses a dye that specifically binds to DNA only, so it is likely a more accurate quantification of our genomic DNA.
Once we quantified our DNA, we designed primers to amplify the region with our mutation, and we ran a PCR experiment. After PCR, we analyzed our DNA using gel electrophoresis to confirm if the desired region was in fact amplified. We expected to see bands around the 804 base pair mark, which is the length of our amplicon. But the gel on the left is a result of our first PCR, which did not successfully amplify our DNA. So we did a second PCR and it still did not amplify our DNA. So we were not able to send any for sequencing.
Since we did not get the desired results from our PCR experiments, we had to analyze the string sequencing results from group PBE, who had the same gene sequence as us. We had some difficulty locating their mutation, but we eventually found it on the reverse complement strand. The C allele, as mentioned before, was the wild type allele. The nonsense mutation we identified involved a C changing to a T, and we believe that group PBE may have mistakenly sent out a wild type sample for sequencing instead of a mutant. And the figure at the top right of the slide is just showing the amount of coverage, and we had high coverage from our Sanger sequencing result.
And then the last part of our mutant analysis was that we looked to compare the phenotypes of wild type and mutant plants. We used thin cross sections of the first internode and stained them with pH HCl and T blue stains. These are all of the wild type plants which show intense red and blue color.
And here are the mutants, which are showing less intense red and blue staining. One of the mutants shown in the bottom left had a dull bluish gray color from the T blue stain, which was not observed in the wild type plants. The differences in color intensity may indicate different lignin and polysaccharide content between wild type and mutant plants.
And then lastly, we set up our own experiment to test the function of our gene based on what we had learned about it. So
since our gene showed differential expression between etiolated and de-etiolated plant shoots, we decided to test the effect of a specific wavelength of light, blue light. We grew 40 seeds in agar plates and wrapped alpha plates in foil to block out all light. Seeds were left to germinate for 4 days. Then the foil was removed from two of the plates, which were then exposed to blue light for 24 hours. We then harvested the shoot growth to extract the RNA and analyze it for any differential gene expression of our gene.
then we extracted the RNA and quantified it with molecular absorbance spectroscopy and analyzed it using gel electrophoresis. All 6 RNA samples in lanes 2 through 7 showed 2 distinct sets of bands that indicate the 28S and 18S rRNA. But as you can see in lanes 2 through 7, there's also some smears, which indicate the actual mRNA, because mRNA transcripts exist in all different lengths, so they will therefore reach different points on the gel.
Next, we used reverse transcription to make cDNA from our RNA samples. We attempted to quantify our cDNA with qPCR and compare the expression levels to that of housekeeping genes, but our experiment was unsuccessful. As you can see here, this is the plate setup that we used, where DET means de-etiolated, ET means etiolated, GOI is the gene of interest, our gene, HKG is the housekeeping gene, and W stands for wild type cDNA provided by instructors, and G is mutant DNA, mutant RNA, sorry, provided by mutant cDNA provided by instructors. So we attempted our qPCR again, and again, no data was found.
So finally, some main conclusions that we can make about our gene is that it's likely involved in the early development of this time. We also know that the amount of light likely has an effect on the expression of our gene, but we can't conclude what type of light since our blue light experiment was unsuccessful. So some future directions could be to just repeat our etiolated versus de-etiolated experiments with blue light, and then also with other wavelengths of visible light.
And of course, our acknowledgements. Thank you so much. And that's all we got.